like this massive regulation. It's like extremely important. So I want to make sure that we got this regulation down. Good? Now, in that chapter, they also conclude, and you can look in your books. Connor, you might want to do that. You can look at the page numbers off to the side. Would you look on, like, maybe 2 or 3 pages later, would you look at the right-hand side? I need you to show me, there's I think it's like maybe 224 or so, where it talks about, like, density independent and then density dependent inhibition. Go a little bit further. One more. Maybe right in there. It's got like a petri dish of cells growing on top of each other, maybe. Yeah. What is that? 226. 229. Page 229. You do want to look a little bit at density independent inhibition, density dependent, like inhibition, like these ways that you can stop growth. Basically, this process is highly regulated by the amount of cells and the thickness of those cells. And what that does is that essentially prevents like uncontrollable growth. Remember, uncontrollable growth is typically the sign of a tumor, which is the sign of possibly cancer cells or cells that could become cancerous. All the difference between that is that cancer cells have a chance of possibly moving or developing into cells that will move and travel throughout the body and maybe create a tumor on a vital spot like common bile duct or ureter or any other thing. Cool. So there's the mitosis chapter. We still have like 10 minutes or so. So what other topics might you have? Yes, ma'am.
The general like sequencing of DNA, like transcription stuff like that. Like what's the order
of replication? Now, transcription translation is all about protein synthesis. That's chapter 17.
Right, but it still involves DNA.
Right. So see, there's the two different ones though, and this is it's very difficult to keep them straight, and it is a task to keep them straight, just because. So, you know, my like when you say like DNA replication, that is not transcription translation. That is not RNA polymerase.
No.
You want to use so the question is, do you want to use the term DNA polymerase and copy the DNA, or do you want to use RNA polymerase and read the DNA? Because those are two different things.
Let's start with the copy.
Yeah.
But now, because I do want to make mention, I want this to make sure that like this is syncing, that because a lot of textbooks, yours doesn't, but a lot of textbooks teach them in the same chapter, and it is an issue for people very commonly to get those two confused. Do you realize I'm in two different chapters in my head right now? I'm in chapter 16, which is to copy the DNA. I'm in chapter 17, which is to read the DNA. Are you going to copy the book, or are you going to read it? Yes, your question. I'll come back to your point.
So the RNA is used to make proteins, right? And we use RNA polymerase.
That's right. That's correct.
And then the DNA polymerase is when you replicate
And make new DNA.
and make
Yeah, so the word polymerase is just a nice way of saying an enzyme that will make a large nucleic acid chain. The question is, what are you making? Are you making a DNA nucleic acid chain, or are you making an RNA nucleic acid chain? And you would use them at different times. So like the DNA chain would be like useful in replicating the DNA. The RNA chain would be useful in like making a protein. So there's a different time and place. Now, as far as like the different events, is that what we're kind of wanting a little bit? Because it will take me a little bit longer. I'm not gonna answer that today, but if you run me tomorrow, or if you guys will write down, that's a topic that definitely we want to cover. Just basically say chapter 16 replication in your notes, and then I'll know that that's where I want to start. There's still time for like there's still 5 minutes, so there's still time for questions. It's just that's a process that will take me like 12 minutes to get through. So like the idea of like different types of cells. Okay, so you probably just want to jot this down. There's different types of cells that plants have. Remember, there was like 3 different organs, there was 3 different tissues, and there was 3 plus 2, so 5 different cells. Remember the plus 2 were like your vessels and tracheids and the like vascular cells. But your question was over the 3 like normal types of cells that plants have. Probably want to write this down: collenchyma, sclerenchyma, parenchyma. Collenchyma, sclerenchyma, collenchyma. This is in chapter 35.
Are those separated by density?
It's really, they're separated by function and actually the way their cell walls grow. And both collenchyma and sclerenchyma the cell walls have an ability to grow of unique thickness on opposing sides or on different sides. So you can have that's kind of the identifying feature of like sclerenchyma and collenchyma, is cells that have a cell wall that's this thick on the left side, but this thin on the opposite side, and you would only have that unique distribution of thickness of cell wall if you were having that cell being used on the out part of the plant, or being used in a growth situation. Because when plant cells grow, they grow heavy on one side to allow it to bend over. So really, the sclerenchyma and collenchyma are support only and structure based. Your parenchyma, and I remember the parenchyma with the P, and then I stick that directly to photosynthesis in my head. That's like, this is all my little tricks to remember how to do this. Parenchyma, P, photosynthesis, and I think it's a metabolism cell. So when we talk about the goo in the middle that's doing the photosynthesis or the respiration, that's the parenchyma cell. 9 times out of 10, actually more like 10 out of 10, if they ask you for that, they're going to ask you for parenchyma, and they're going to want you to put it in the light of photosynthesis and it being a metabolism-based cell. Still more time for questions, yes?
Well, do you think tomorrow we can also talk about like photosynthesis and like the chain, you know, like
The light reactions versus the...
Yeah,
Yes,
stuff.
so jot that down. That tomorrow we want to talk about we want to talk about photosynthesis, chapter 10, and we want to talk about chapter 16, replication. So I'll throw out some other topics to you, and you guys, you can write these down if this triggers something. You guys definitely want to be asking questions. The big top, another big topic is genetics. When I use the word epistasis versus incomplete dominance versus polygenic inheritance versus pleiotropy, are all those terms set or are all they not set? That right there is going to be at least like a small 4 or 5%, those terms just alone. Hardy-Weinberg equilibrium, there's another one. What's the chance of that showing up? That's pretty definite. Chi-square, unlikely to show up, but maybe if you want to go over it, we can. And test to see if your data is valid or not. What else is there? Let's see. You had said photosynthesis. I would assume that respiration's okay. If that's an issue, is that an issue for anyone? Do we want to go over respiration at all? Or do you want to see what the test says? I could draw out the picture of the mitochondria, like I do.
Maybe just like a quick order.
Okay. So maybe something quick about respiration.
And
The enzymes involved, okay. How about cell parts, organelles? Those should be okay. Lysosome, digestive organelle, peroxisome. Takes H2O2 and makes water from it. Everything else is pretty much set. Remember the worst one to remember is the smoothie R, its functions. Remember what your smoothie R is doing? Okay. Come on, guys. What is this? Smoothie R is doing 3 things: detoxification, calcium storage, and lipid synthesis.
And how does the smoothie R stay inside the cell and not go outside?
No. The ribosomes that are free, proteins stay inside. Ribosomes on roughy R, proteins are going to go outside. We still have like 1 minute, enough time for like one more question.
What is biology?
What?
What is biology?
Stop, Joseph.
Study of life.
What day is our actual EP test?
It's Monday, I want to say like around the 13th or so.
It's the 14th.
The 14th.
Okay.
Okay, well, that can conclude today, I guess.